Hi, everyone. Good morning, good afternoon. We're going to be starting in probably about 20 seconds. Good morning. Farhan, can you hear me okay? So thank you everyone for joining us today for this webinar. We're really excited to have it. So the title is Biomarker Discovery in Custom Panel Development for Multiple Sclerosis Disease Assessments. So this is utilizing the Olink platform. So my name is Arthur Chung, and I am the Business Development Manager here at Olink and the host for today. So a little bit about Olink. It is a protein biomarker discovery company that is headquartered in Uppsala, Sweden. It also has US headquarters in Boston. So Olink, in essence, gives you greater insight into the biology of what you're studying. And this is in the context of basic research, as well as translational research, or in a clinical trial setting. So the core of the technology is called proximity extension assay, so PEA for short. And what it does is we rely on two antibodies that bind to a target protein. And when those binding events happen, you get a readout. And so in essence, this is what drives the sensitivity and the specificity of the assay. We've got panels that allow you to target anywhere from 45 to almost 1,500 proteins at a time. And we have a service whereby when you send us your samples, we'll be able to run it within weeks and not months. And the data that you get back is reproducible, it's high quality, and it really is backed by outstanding service, support, and the expertise that we have in both applications as well as biostat. So I am proud to introduce Farhan Qureshi. He's our speaker. He is the VP of Biomarker Product Development over at Octave. He received his bachelor's in molecular and cell biology from the University of California at Santa Cruz, and he has over 20 years of biotech experience in the Bay Area. So some of these roles include at Genentech, Avere, Crescendo, and now at Octave, he leads cross-functional teams involved in R&D to support various therapeutic development and areas, so including IVD. So without further ado, I'd like to introduce Farhan. Go ahead, Farhan, take it away, all yours.
I appreciate the invitation from Olink to present this webinar, and I also want to thank and welcome everyone who has joined the webinar online. Here's the topics that we'll be covering today. So I'll start with a background both on multiple sclerosis and the Octave Care platform. Next, I'd like to discuss the selection process that we use to include biomarkers onto our custom assay development project, and I'll walk through that project. I want to highlight some of the results from our analytical validation studies that we performed at Octave, as well as highlighting some of the results from the biomarker studies that we performed with this custom panel. I'll then discuss next steps and summarize before we move into a Q&A. So background, this is the problem with which we are trying to address at Octave Bioscience. So multiple sclerosis is a chronic debilitating disease. It affects nearly 1 million patients in the United States, with a 3-to-1 ratio of women to men. The onset is on average in the late 20s. So this being a lifelong disease, it's a disease that patients are dealing with for decades after diagnosis. It's also a progressive disease driven by inflammatory processes, immune modulation processes, demyelination, and neurodegeneration. The disease burden leads to 50% of patients being on disability within 12 years. There's a very diverse therapeutic landscape with over 18 approved therapies with annual costs of $60,000 to $80,000. They have varied efficacy and safety profiles, and there's generally an inverse relationship there where drugs that are more efficacious have higher risk factors. 3 to 4.5 million in lifetime costs per patient to manage the disease. So in summary, it's a high cost disease. There's significant variation within the population and generally poor outcomes for patients. The current tools are subjective, qualitative, and inadequate. And so new measurement tools are needed to expand clinical insights and improve outcomes. And that's where Octave comes in. To improve outcomes, we need to transform measurement and management of the disease. So at Octave, we are using serum biomarkers, enhanced MRI insights, and patient tracking tools that are being integrated via what we refer to as a care platform. And this care platform takes these layers of data, including others, that are factored into protocols and decision support tools to address significant challenges, issues, and unmet needs across the entire MS ecosystem. And by ecosystem, I'm referring, of course, to the patients, to the physicians, to the drug developers, and also to the health plans. And the goal in managing MS patients is to manage to NEDA, or no evidence of disease activity. Also avoiding relapses and slowing the progression of the disease, and help guide the use of disease-modifying therapies. So before I talk about our biomarker program, I want to cover our enhanced MRI insights program and our patient tracking program, which are also part of our care platform. So for our enhanced MRI program, we are dramatically improving an existing modality to deliver new insights. So MRIs are currently used as part of the standard of care to monitor patients who have MS. Typically on an annual basis, MS patients have an MRI. And we're using machine learning to quantify, provide serial reporting of longitudinal scans with expert reads and interpretation by neuroradiologists who are focused on MS. The reports that we provide are contextualized to the use case and also clinical objectives, and we make both the images and the reports available to share at the point of care. The goal is to improve the quality, decrease ambiguity, and increase clinical actionability for this existing modality. For our patient tracking program, we've developed a patient mobile tool, and this mobile tool allows for specific therapeutic compliance tracking. So as an example, patients with MS are often on injectables that they have to administer multiple times throughout a week. And so the ability for a patient to track that, also symptom tracking and relapse screening capabilities. And notably, our patient mobile tool has direct interactions with a care partner who's an MS certified nurse that interacts directly with the patients. We also provide reporting and alerts to the patient's care team during their care cycle. And by care cycle, I'm referring to all of that time that occurs between their doctor visits, which is typically 6 months. We're also in the process of incorporating passive monitoring capabilities via sensors and wearables, and this will represent a critical new data stream for physicians to manage their patients between their visits. So on to our biomarker strategy. Octave is creating novel proteomic tests. By measuring proteins, we're able to get a real-time assessment of the biology, pathways, mechanisms, and cell types that are involved in the pathophysiology of the disease. Proteins also enable a dynamic quantitative measurement of these pathways, which include inflammatory pathways, immune modulation, demyelination, and neurodegeneration. And a biomarker test can address several issues, including monitoring of subclinical disease, detecting relapses, monitoring progression and silent progression, and even therapy response and selection. The idea behind a biomarker test is that it gives a more sensitive, dynamic measurement of the biology, which can then lead to earlier insights and thereby enable faster interventions. There's two concepts when we think about MS disease activity and disease progression that I want to describe. So disease activity is the symptoms that occur that manifest as relapse, flares, or exacerbations, and disease progression is the increasing level of physical disability that a patient experiences throughout the disease course. And we've partnered with Olink for both the research phase, where we access their RUO panels to conduct deep scans, and to manufacture a custom panel for development and validation at Octave. So this diagram represents how we see the current standard of care. And this is a very complex and heterogeneous disease, and MS pathology is poorly captured with these current tools, the MRIs and the clinical assessments that are currently available. This prevents a holistic and prospective approach to managing the patients. By using a biomarker test to provide a real-time assessment of the underlying biology, these can be utilized alongside enhanced neuroimaging and clinical tools that I described as other two data streams that are part of our care platform to then enable prospective management rather than reactions to damage. So again, earlier insights can lead to faster interventions. Our biomarker tools test are focused primarily on disease activity to start, but also neurological damage and the association of biomarkers with several pathways beyond inflammation, including neurodegeneration, immune modulation, and myelin integrity, allows us to provide a greater depth of information. Market research. So before we starting our biomarker development program, we conducted market research with KOL neurologists as well as community neurologists that confirmed two target product profiles. Both the disease activity test and a disease progression test generated high interest and had unique attributes with the potential to meet differing unmet needs. And both of these test concepts scored extremely high on the ranking scale that was established. The top three attributes for our disease activity test were identified as accurately identifying patients in remission versus those who have subclinical disease, demonstrating whether a therapy is having a favorable effect on disease, and identifying patients as early as possible in the flare cycle. The top three attributes for the disease progression test were providing early evidence of disease worsening at the subclinical level. A multivariate approach would be applicable both to current therapies, considering the mechanism of action, as well as new therapies that halt progression and potentially regenerate or restore function, and also identifying those patients that would be needing more advanced therapies for intensification. So this research provided further confidence and guidance to us, to our team, that we were developing solutions that would add value to the field. We acknowledge that our mission would not be easy. So there's several aspects that are applicable to biomarker work in general, and some that are unique to MS that make this sort of process very challenging. And we've kept these at the front of our mind as we've continued through our development of our biomarker tests. And some of these factors that have impeded progress are, as I've discussed, the complexity and heterogeneity of the disease. Many different pathways are involved. And there's also a wide range of potential confounders and comorbidities. So these patients aren't just dealing with MS. There's other comorbidities that can factor into the biology of the patient. There's a high level of heterogeneity, both within the population. So there's several defined phenotypes. Although there's a trend towards recognizing MS more of a spectrum, there are still defined phenotypes with differing biology, and also intra-patient heterogeneity, where as the disease progresses in a patient, their biology can evolve. The endpoints that we use can be subjective and qualitative and variable. So when we are using protein signatures to hold the ground truth for which we are developing our algorithms, we have to acknowledge that some of the endpoints are subjective and qualitative. Many of the studies are underpowered and only performed in single cohorts. There's also insufficient pre-analytical handling, which prevents looking at cohorts or blending cohorts for a combined analysis. There's also a scarcity of longitudinal multimodal data. So studies for which we have longitudinal serum samples along with richly annotated clinical and radiographic data are few and far between. There's been a heavy focus on univariate approaches and limited application in multivariate statistics in the modeling that's been done. And there's also been setbacks in transitioning from research to clinical grade assays. So these are all factors that have impeded progress and that we've thought about deeply as we've developed our biomarker test. So why Olink? So many of us at our team at Octave have worked with several immunoassay platforms, both at Octave and before, and all of them have unique attributes and advantages. Olink had several that led us to partner with them for this project. So one of them was their extensive library that enabled us to cast a very wide net in our discovery studies. So on the Olink platform, we accessed over 1,200 analytes. And as many of you know who are familiar with the platform, we can do that with minimal volume requirements. And the samples that we had the fortune of access to through our collaborations were very valuable samples, precious samples. So minimal volume requirements is a great attribute. With that wide net available in their library, that included key content that was of extreme interest to us, and that was also focused on disease and biological pathways that were highly relevant to MS. PEA technology that Arthur described in the beginning also addresses some of the specificity issues that can be common in high multiplex immunoassays. And additionally, the qPCR readout offered sensitivity in serum that was equivalent or better than conventional immunoassays. We leveraged Olink's service offerings for our early studies, and we also obtained training and certification from Olink to perform the off-the-shelf R&D assays in-house to maximize throughput and gain experience as we develop the custom assay, and we're going to bring that analytical process in-house. There was also a very collaborative approach to executing a custom assay development project. This included optimizing performance of existing analytes with absolute quantitation, so reporting out in picograms per mil, nanograms per mil, etc., adding new assays, the ability to add new content, and then thorough characterization of performance per stringent specifications that we had established, and manufacturing multiple lots that we could then take through an internal analytical validation at Octave. So a little bit on how we selected the biomarkers for our program. So our process to select biomarkers was staged into feasibility, discovery, development, and validation, which is the stage that we're currently in now. As we advanced through each stage, we were increasing the number of samples and the studies we had access to, for which we used to hone in on the features that would be included on our custom panel. We were very fortunate to gain access via collaborations to several deeply phenotyped studies in which we had serum samples that had been collected alongside a rich set of associated clinical and radiographic features. This also enabled us to define which endpoints were most relevant to disease activity assessments. And the process by which we used to select the biomarkers that were on our panel was dynamic and iterative. And so the maximum number of analytes that we had measured was over 1,400. This was not just on the Olink platform. There were additional platforms that we accessed. And we went through an iterative process where we successively honed in onto the number of biomarkers that we would include on the final panel. And the criteria that we used incorporated both correlation and causation. And so this was the process that we went through. We first would conduct exploratory data analysis upon completing a biomarker study. That included analytical QC checks, so looking at accuracy of controls, performance of the assay, precision, and in the case where we had multiple biomarkers on different platforms, we could look at cross-platform performance for consistency and correlation. Also exploring the most appropriate transformations for both the analytical data that we were receiving, as well as the clinical labels that we were using to associate with our biomarker profiles. The next step was to perform univariate data analysis. And so we set some optimization metrics, such as area under the ROC curve, p-values, that would allow us to interrogate our biomarker performance at a univariate level. Another important aspect of our univariate process was to look at demographic relationships. So along with the protein concentrations and the endpoints, we also had several demographic categories, such as sex, age, disease duration was another one, and several others as well. And so we look for relationships of the biomarkers with these demographic factors independent of the endpoints. And then we also perform correlational analysis, so we can look for collinearity amongst the features at a univariate level, leading into our multivariate model building strategies, in which we want to leverage the power of orthogonal effects. So investigating collinearity is important to do at the univariate level. For our multivariate model building strategies, we perform feature selection, dimensionality reduction, and forward selection. And based on the study characteristics, we applied cross-validation techniques to ensure generalizability without overfitting our models. And this included leave one out cross-validation, k-fold cross-validation, up to training models in independent studies and testing them in an independent cohort. This was combined, the univariate and multivariate feature importance, with biological corroboration and also analytical performance. So I mentioned that we started by looking at the performance of the assays from an analytical perspective. So we combined these four considerations to identify the 21 most compelling proteins. This is a list of the 21 proteins that are on our panel that were manufactured onto our current panel. I've also included some of the backup markers. When we selected the features for our biomarker panel, we also included some backups, and I'll describe how those came into play. So I have the marker name here, as well as the alias, and some of the associated pathways and cell types with these various biomarkers. And so how did we identify those pathways and cell types? So as we went through our biomarker studies, features that were statistically relevant were investigated for their association with MS pathophysiology. And we use several methods to do this. We use computational models, automated and manual curation of open source ontologies. We built a probabilistic graph network to aggregate the findings in the literature. We access string and cytoscape for protein-protein interaction modeling, gene set enrichment for functional annotations, and also spatial expression profiling. So this was our process that we went through to identify the key pathways and processes that were associated with these biomarkers that had shown to have strong statistical relevance to our endpoints. And there were several key pathways and processes that we found our biomarkers represented that were known to be associated with MS. And there was a lot of work that went into this process, and we tried to summarize it on this diagram here, which shows 10 key hallmarks of the disease on the outer rungs, along with subprofiles for each of those hallmarks. And for each one of those subprofiles, we've associated our individual biomarkers on the panel with those profiles. On the captions on the outside of the diagram, those are radiographic features that we either have or we will be associating pathway profiles, so using restricting models specifically to subsets of biomarkers that are associated with these pathways or these subprofiles. And so we can use different features, radiographic features, to help us interrogate pathway-specific models, in addition to looking at the overall disease activity score. So our custom assay development project. So the process that we went through was first to select analytes, and that included several backups as mitigation for the panel development. We defined specifications for the development stages, finalized a project plan in collaboration with Olink, and established timelines for this project. We then procured reagents and raw materials, and this was to support 2 lots that were manufactured, and the intent was to vary the raw materials and reagents to the extent possible so we could show equivalency and bridging between lots. We also provided MS sample panels for characterization to Olink at this point. So we had internal cohorts for which we provided large volume of samples from our intended use population to Olink for them during their characterization process. We then did a technical feasibility process that was performed at Olink and reviewed by Octave. An intermediate panel development stage then occurred where we added a new assay and also performed a pilot lot manufacturing so we could look at a multiplexed pilot lot of these 21 analytes together. When that stage was completed, we went through with final panel development. We manufactured the 2 lots. Analytical characterization was performed by Olink. Lot bridging was also investigated to maximize the equivalency. Stability studies were performed in order to look at stability of both reagents and samples, and we optimized the NPX Manager software for the application of our assay. Next, an analytical validation was performed at Octave after transfer of the reagents. We then reanalyzed all of the feasibility studies that we had previously tested using the RUO assays and our custom assay, and we're now in the process of conducting a clinical validation study at Octave, which is looking at several independent cohorts in our custom assay. So a little bit on the technical feasibility stage. Our goal was to develop a 21-plex panel that was performed in serum samples at a single dilution. So we wanted to plex this all onto a single assay. And so the technical feasibility analysis was done by looking back at our previous data from the RUO assays. So these are the 92-plex assays that are available off the shelf from Olink to identify analytes that would require dynamic range optimization so that they could all be plexed into a single panel at a single dilution. And we identified several analytes that required dynamic range optimization. And so the goal is that you want your sample response to fall within a linear portion of the curve. You don't want it too close to the bottom of the curve or too close to the top of the curve. So when we went through this optimization process at Olink, this involved reagent modifications as well as investigating some of the source material that we were using for the absolute quantitation. 7 of the 9 analytes for which we attempted dynamic range optimization were successful. And this was the point where we identified biomarkers, and I have a couple examples on the bottom, where in spite of dynamic range optimization, in both these cases, the sample response was too close to the upper asymptote of the curve. So we decided to drop those markers and replace them with backups. When we made those decisions on replacements, we incorporated analytical, statistical, and biological considerations. So we went back to that process that I described earlier to decide which biomarkers were most appropriate to replace these. Another important step in the assay development process was the development of a novel assay. And so GFAP is a protein that is expressed by numerous cell types in the CNS, including astrocytes. In our studies and in the literature, it's shown relevance to MS disease endpoints. And this was an assay that was not available previously on the Olink platform in the RUO assays. So this required an assay development project. So Olink developed this process. We accessed reagents, and we first looked at pilot data to determine that the assay had met some preliminary specifications. The assay performed well in the pilot data, and we then included it in the final 21 for our production lot manufacturing. So Olink then went through the process of manufacturing and characterizing 2 reagent kit lots at Olink. When the characterization was done and reviewed by Octave, they transferred the reagents to us for us to perform the analytical and the clinical validation steps. Just an important point about the custom assay panel and absolute quantification. So those of you who are familiar with the Olink panel, likely many of you are using their really powerful RUO off-the-shelf panels. And those report out in normalized protein expression. And so you have to go through some extra steps in order to bridge studies. And for our custom panel and for our intended use, it was very important that we did not have to go through those additional steps in order to look at consistency of results between batches. So therefore, we designed the panel to have absolute quantification. And what that means is that multiplex standard curves were prepared along with 3 levels of calibrators for each one of the analytes to cover the range of the sample response within the MS population. And I've shown an example here for one of our analytes, which is myelin oligodendrocyte protein, where we used the internal MS cohort that I discussed earlier, and we established what the minimum, the median, and the maximum was of the MS population. And then the calibrators were designed to fall within the linear range of the standard curve, and additionally to cover the sample response that we saw in our intended use population. So we went through this process for each one of the 21 biomarkers on our panel. The reference standard curve, a 4-parameter logistic regression, is reestablished for each lot of the reagent kits during the manufacturing process of that lot. And then the calibrators and a blank are run on each assay plate. And the signal obtained from the assay in normalized protein expression units is converted back to absolute concentration using the calibrators referenced back to the gold standard curve. So this was a very important component of our custom assay panel, and one in which Olink did a great job of meeting our specifications in terms of designing an absolute quantification approach that represented our intended use population in MS samples. So I want to highlight some of the analytical validation results and experiments that we ran once the assays were transferred to us. So this is the process that we went through, and we completed this process using the 2 manufactured lots or batches of reagents that we received. So a dual lot approach and a dual site approach as well. So the characterization first occurred at Olink, and then this was followed by the analytical validation at Octave. We investigated the following specifications: accuracy, and we did this using sample mixing experiments, so we could evaluate this for endogenous protein, as well as looking at correlation to the R&D assays, or the RUO assays that were used for our feasibility studies. Precision, we characterized both intra and inter-assay precision for the individual biomarkers. We looked at sensitivity, established limit of blank, limit of detection. I'm going to focus on some of our lower limit and upper limit of quantitation results. I think those are the highest stringent specifications that you can apply to an immunoassay. Interference, we looked at heterophilic antibodies. We looked at common endogenous substances that are routinely investigated for interference, and also drugs as well as specific MS therapeutics, or DMTs. We investigated cross-reactivity. This included intra-panel, so crosstalk amongst the 21 analytes, as well as homologous proteins. We characterized stability, so we performed both accelerated and real-time stability experiments for both reagents and samples, including looking at the number of freeze-thaw cycles for samples. We characterized robustness by varying both the equipment, the personnel, and the laboratories in which the experiments were run. We performed an initial diurnal variation study where we looked at variability over 6 time points in a relatively short duration of time, so across 12 days. I'll share some of that data. And we established reference ranges, and we did this both for healthy controls as well as our MS intended use population. We've also defined a process for kit qualification and release, which is a multi-stage process conducted both at Olink and Octave, and it includes several factors, including correlation, accuracy, sensitivity, and precision. And notably, this is done with patient samples, I should say endogenous serum samples, so that we are looking at the output of the assay relative to endogenous protein as opposed to recombinant protein. The protocol has been designed with a high level of quality control. So we have 3 levels of internal controls for each sample, as well as 4 process controls, which are serum pools that are run on each plate. We've established specifications for accuracy and precision of our calibrators, our controls, and our samples using the internal controls that are loaded in each well. So accuracy. Accuracy was determined by mixing serum samples at different ratios and then evaluating the % recovery of the observed concentration relative to what the expected concentration. And I discussed the advantage of doing accuracy this way is that it can be performed using endogenous protein. The expected concentrations are calculated by applying the targeted ratio of the unmixed samples. So the samples are run unmixed as well. This provides for 20 mixtures of samples that we evaluated for each biomarker. And in the table on the upper right, I've indicated what the minimum and maximum concentration that we characterize accuracy for each of the 21 analytes. We established an acceptability criteria of 80 to 120% recovery, and all analytes passed this specification. All of them were within 90 to 110%, in fact. Next, I wanted to cover some of the work that we did to characterize precision. So intra-assay and inter-assay precision were characterized for each analyte. We did this by calculating the % coefficient of variability or variation determined using serum pools. And again, this gives us an opportunity to characterize precision using endogenous protein. As we went through these experiments, the equipment, the reagents, and the location were varied to demonstrate the robustness of the method. And these serum pools that we've used represent different populations. And so 2 of them represent MS pools, so pools of serum from multiple sclerosis patients. 1 is an rheumatoid arthritis pool, which serves as an inflammatory disease control, and 1 is a healthy control pool. We established an acceptability criteria of less than 15% for intra-assay and less than 20% for inter-assay CV. In our intra-assay precision experiment, we performed 12 replicates for each of these 4 serum pools that were analyzed within a single plate. And our inter-assay precision assessment was analyzed per process control across 51 plates. And this was done across 2 different lots, so up to an N of 51. 20 out of 21 analytes passed the specifications that I just mentioned. One of the analytes, call4A1, was unacceptable in terms of this specification. So intra and inter-assay % CV were higher than the specification. We had observed some of this imprecision during both the characterization as well as some of the initial experiments that we received when we got the reagent lots from Olink. But we continued to assess this biomarker, and based on these precision results, we've decided to exclude this biomarker in our modeling for MS disease assessment associations. So essentially, we have 20 biomarkers at this point which have passed our analytical specifications. I wanted to highlight some of the results we did to characterize sensitivity. So we manufactured a limit of quantitation panel that was evaluated, and this panel included 4 levels each near the upper anticipated and lower anticipated limits of each assay. And the way we targeted those anticipated limits was based on the shape of the standard curve and the location of the asymptotes. These LOQ panels were run in triplicate over both lots, and there was a minimum of at least 5 runs per lot, and they were then fit back to the standard curve to determine the concentration in picograms per mil. We established an accuracy and a precision specification, and these were used as acceptability criteria to establish both the upper and lower limit of quantitation in the assay. Those are represented on the table above, the LLOQ and the ULOQ. In addition to that, we also established reference ranges in the MS population. So we analyzed 928 serum samples during the assay development process, and we used those to establish our MS reference ranges at the 2.5th and the 97.5th percentile. And we then went back and looked at that population of samples to determine the count and percentage of instances where the concentration was outside of our LOQ ranges. And we found that the assays were highly sensitive, and the maximum percentage of samples that would require imputation at any LOQ was 2%, and that was for the biomarker CDCP1 at the lower limit. And so what I mean by imputation is if we run a sample and we determine that it is either below the LLOQ or above the upper limit, we then impute to that established lower or upper limit, and we use that feature, that value as the value that is inputted into our algorithms. So assay interference. We did extensive work to evaluate assay interference, and this included looking at therapeutics, both MS disease-modifying therapies as well as common drugs. We looked at heterophilic antibodies, so rheumatoid factor and human anti-mouse antibodies that many of you who are in assay development know that those can pose problems for multiplex and even singleplex immunoassays, and also endogenous interference. And we used a slightly different approach for each one of these. So to evaluate the analytical interference of therapeutics, we used our serum pools, and these were spiked with the MS disease-modifying therapies and the common drugs. For the MS disease-modifying therapies, those that we had access to were tested at concentrations that were targeted at 2 times the reported Cmax, using the PK results from the literature, or in some cases, the highest possible concentration that was obtainable via our spiking stock. And then for our common drugs, we based that on the CLSI standard, the components that we tested, as well as the concentration. For RF interference, rheumatoid factor, we accessed a RF concentrate that we then used to spike into serum samples at both high and low concentrations. For human anti-mouse antibody interference, we accessed samples that were confirmed positive for human anti-mouse antibodies. And then we did a similar experiment to our accuracy experiment where we mixed them with MS samples from our internal cohort. For endogenous interference, I'm referring to bilirubin, hemolysis, and lipemia. We used our 4 serum pools and spiked those at typical concentrations per the CLSI standards. We then looked at percent recovery, whether it was relative to the spike controls or relative to the expected concentration as determined from unmixed samples. That's pertaining to our human anti-mouse antibody experiment, similar to the way we did our accuracy experiments. We established an acceptability criteria of 80 to 120% across all the spikes or sample mixtures to characterize the level of interference from any of these investigations. In this case, we saw that for one of the biomarkers, COL4A1, there was under recovery for several of the drugs. And if you recall on an earlier slide, I presented that COL4A1 was a biomarker that had imprecision. So we believe these results are likely an artifact of the assay imprecision. And as I mentioned, we're not including this biomarker in our modeling relative to MS endpoints. There was one finding for the biomarker CCL20. Cefoxitin spiked at a high concentration resulted in a percent recovery that was slightly below our acceptability criteria. So we're going to further characterize that in follow-up experiments. We plan to test additional DMTs as well, MS DMTs, and so we'll include a follow-up of this in those experiments. For all the other assessments, the median percent recovery was observed to be within our acceptability criteria. Diurnal variability. So this was a study that was performed so we could characterize fluctuations that can occur in biomarker levels between days over a relatively short duration. So this initial study consisted of 8 subjects with 6 time points. So day 1, 2, 3, 4, 5, and 12. For each time point and subject, we calculated the percent difference of the observed protein concentration relative to the average concentration that was determined for all 6 time points. So that's the data on the graph on the upper left. Additionally, we looked at this percent CV for all 6 time points for each patient. The mean and median percent differences for each biomarker were observed to be within plus or minus 20%. That's the graph on the left. The mean and median percent CV was found to be less than 20% for 18 of our 21 biomarkers. And so I'll talk through the 3 biomarkers that didn't meet that specification. We did observe a higher level of diurnal variability for COL4A1. Again, that's the biomarker that's likely being impacted by the established assay imprecision. And CCL20 was a biomarker for which we observed percent CVs to be greater than 20%. We're going to characterize this result further in a future study that I'm going to describe that we're currently in the process of designing. Growth hormone results were found to be more variable than the other 20 biomarkers. The growth hormone is a biomarker it's well established in the literature to have a high degree of both ultradian and diurnal variability. So even within a day or between days on a short duration, well established to have a high degree of variability. We're designing a follow-up study restricted to the MS population that's going to include multiple draws within a single day, as well as additional time points beyond 12 days. So we'll be further characterizing both diurnal and ultradian variability in our custom assay. So that's a summary and some highlights of the analytical validation result. There's several other experiments that we've performed. And at the end of this presentation, I've provided a link to several of our presentations that we've done at conferences, ACTRIMS and ECTRIMS, including a thorough presentation on the analytical validation results. So now I'd like to discuss some of the biomarker associations that we've seen in the studies that we've performed. So this is just a summary of univariate performance for custom analytes for disease activity endpoints. This is a univariate summary, and so the biomarkers that are highlighted have shown significance with a p-value threshold of less than 0.05. These are the endpoints that relate to disease activity, and as I move through the next slides, these are the endpoints for which I will show multivariate performance as well. So I just want to speak about the endpoints. So GD stands for gadolinium, and this is the classification of the presence or lack thereof of gadolinium enhancing lesions on an MRI that was administered within close proximity to the serum draw. So gadolinium is a contrast enhancing agent that's administered in some MRIs that then can cross a disruption in the blood-brain barrier to be indicative of radiographic evidence of a relapse. So we resolved on using gadolinium enhancing lesions as our primary endpoint for our quantitative assessment of disease activity in patients. I've also included a category which is referred to as gadolinium shifts. This is where we've used longitudinal results. So rather than using the absolute concentration at any point in time that a biomarker result was determined, we use the shifts in previous time points for which we also have biomarker results. So instead of using the concentration, we're using the shift between 2 successive time points. And we use that as a feature that's entered into our models. Relapse refers to the classification of physician-defined relapse status. So at the time that the blood was drawn, was the patient in a state of exacerbation, relapse, or quiescence, remission? And then annualized relapse rate is another endpoint that looks at the rate of relapse per patient and annualizes that over a single year. Here's some of the results relative to our primary endpoint, and these are results that we presented at the MS Virtual 2020 conference recently. And here we're looking at classification and regression results of multivariate models restricted to analytes on our custom panel relative to that gadolinium lesion endpoint that I just described. And so we've looked at both the presence, so whether there is any lesion or not, and we've also used the count to establish categories of disease activity. And so I'll describe those briefly. So subtle disease activity is the comparison between no gadolinium enhancing lesions on the MRI versus only a single lesion. General disease activity is 0 lesions on an MRI versus one or more lesions. And extreme disease activity is 0 lesions on an MRI versus 3 or more lesions. And the optimization metric that we're presenting here for our classification performance relative to these 3 disease activity categories I described is the AUC. And as you can see, as the level of disease activity is increasing based on the radiographic evidence, our AUC of our biomarker model is also increasing as well. So 0.732 for subtle disease activity classification, 0.82 for general disease activity classification, and above 0.9 for extreme disease activity classification. We've also looked at this as a regression problem. In this case, we clip the number of lesions, the maximum number of gadolinium lesions at 5, and we're able to estimate using our biomarker models the number of GAD lesions on the associated MRI. 2 other endpoints that we've looked at are classification of clinical relapse status and annualized relapse rate. So in this, on the left-hand side, this is classification of samples from patients in a state of exacerbation versus quiescence. And so we are able to classify that with an AUC of 0.835, and this is using an 8-feature model to classify those 2 categories. For annualized relapse rate, we set a threshold of less than or equal to 0.2 annualized relapse rate versus greater than or equal to 1 annualized relapse rate. And then we look at our ability to classify those 2 categories of patients. And we were able to classify that with an AUC of 0.737 using a multi-feature model as well. I had spoken about biomarker shifts when I was referring to our univariate results. And so I wanted to highlight some of our data where we've also taken that approach in our multivariate results. And so in this case, we're classifying patients from longitudinal sample pairs that either have increasing or decreasing gadolinium lesion burden. So the patient is either moving from a state of having 0 lesions on their associated MRI to having 1 or more, or vice versa, where they have 1 or more lesions on the first MRI and 0 on the next MRI. And so then we're able to take the biomarker levels at each of those 2 time points on an associated serum sample, and rather than using the absolute concentration, we use the calculated delta for those biomarkers to see, can the shift in the biomarker level provide association with this endpoint? And we see, in this case, even stronger performance than using the absolute concentration. So in this experiment, we trained the model on an independent cohort that consisted of 93 pairs of samples, or 186 total samples. So that model was trained on an independent cohort. And then we took that same model and tested it on a subsequent cohort, which consisted of 73 pairs or 146 samples. And using AUC as our primary optimization metric, we were able to see performance of above 0.9 AUC. And there's several other metrics that we look at as well, including accuracy, sensitivity, specificity when we characterize the performance. I've focused primarily on AUC as I've discussed multivariate performance. So what are the next steps for this project? So currently a clinical validation study is underway. This includes analysis of over 600 multiple sclerosis samples, both retrospective and prospective samples from 4 different sites. We're using the association with gadolinium enhancing lesions as our primary endpoint, but we're also including several secondary and exploratory endpoints for both disease activity and disease progression assessments. When we finalize the algorithms, we will then analyze them in our completed analytical validation experiments to establish performance at the algorithmic score level. So the results that I shared earlier was looking at performance of our algorithms at a univariate, at the individual biomarker. So we'll also evaluate our algorithm scores for analytical validation performance specifications. We're going to further explore expansion of the custom panel's utility as well via collaborative studies, so we can further investigate disease progression, risk prognosis, and therapy response monitoring. And as we go through that process, we plan on incorporating additional biomarkers as needed. We believe that the custom assay panel gives us a good start to investigate additional utilities beyond disease activity, but we certainly see on our roadmap incorporating additional biomarkers, the ability to develop additional custom panels based on unique use cases or unique utilities. So in summary, the lack of quantitative and objective measurement tools represents an unmet need for the MS ecosystem. The disease activity test was developed via a process that involved analysis of over 1,300 proteins in deeply phenotyped cohorts, and we did that relative to several clinical and radiographic endpoints. We used statistical, analytical, and biological factors to iteratively select 21 proteins for inclusion onto our custom panel. Combinations of biomarkers that survey key pathways in MS pathophysiology are able to accurately assess current disease activity in the serum samples. And this disease activity test, our enhanced MRI insights and patient tracking tool programs are being integrated into a comprehensive care platform that will enable a holistic view over time. We plan to link those to care pathways with decision support and a dashboard. So I've included a link to the Octave presentations where much of this data was drawn from. I understand in some cases I moved through it swiftly. In the interest of time, there was a lot of material to cover. So I would hope that those of you who have interest in a lot of the great work that this team has done would reference the links on this site. And I've also included my contact info for anyone who's interested in discussing this project further. We're going to move into a Q&A. I just wanted to take an opportunity first to thank several people. So I should say groups. So I want to thank Olink for all of the support and the collaborative approach in taking us through this process collectively to develop the custom assay. It's been a great experience working with Olink very closely on this project. We have an outstanding lab team and a data science team at Octave Bioscience. I could call people out by name, but I'll just reference them by team. In addition to the wonderful collaborations that we've had with several groups to access these deeply phenotyped studies that enable us to perform these associations with our biomarker signatures. Thank you all.
Thank you, Farhan. That was that was definitely very, very interesting, and I have to say the respect is mutual on our end towards you and Octave. So we have quite a few questions here, and I will just go ahead and start. So many people are you for a very informative presentation. So one of the questions is, I may have missed it, but how many of the 20 analytes did you measure using an alternative technology immunoassay such as ELISA or Luminex? Can you comment on the accuracy of the quantification with Olink compared to another method?
Yes. Okay, so I think there's two questions there. So several of the biomarkers that are on our custom assay had been measured on other platforms as well. I think they mentioned Luminex. Luminex was one of, a Luminex-based platform was one of the earlier platforms that we utilized for our initial study. So before we expanded to the deep scan that we did over 1,000 biomarkers with Olink, we were using a Luminex platform. And there was a good deal of overlap between the proteins that were on the Luminex platform and Olink. There are biomarkers that are on our custom panel that were also on the Luminex platform. And there's biomarkers on our panel that are available via routine ELISAs and several other platforms as well, whether they're multiplex or singleplex. For some of those key biomarkers, we've definitely looked at correlation relative to some of the gold standard assays and characterized that as well. What was the second question, Arthur?
Let me see. Let me go back to it.
Sorry.
Yeah, it's just the question says, can you comment on how accurate the quantification is with Olink compared to other methods?
Yeah, I think I
you
addressed
touched
that.
on.
Yeah.
Yeah, exactly. So let me move on down. So one of the other questions is what is meant by pathway models when you use that term?
I see. So by pathway models, what we mean is we're developing an overall disease activity test. And so by using any of the proteins that are on the custom panel, we are establishing a disease activity score that then reflects the primary endpoint in our study. In addition to that, we are also looking at subsets of biomarkers. So I talked about some of the causation analysis that we had done, and then we are able to categorize biomarkers into some key pathways that are involved with the disease. And so we can restrict the models to only those biomarkers that are associated with those pathways. And that gives us an opportunity to investigate pathway models beyond an overall disease activity model. So it's a restricted set of biomarkers based on our biological corroboration of those of the biomarkers association with different pathways.
Gotcha. Another question is let's just go ahead and read it. Really well described method for iteratively selecting and validating biomarkers that work well together with maximum information content needed to preserve utility in a research environment. So let me see, phenomenal interaction and variability testing. How is enrollment going? Would connections to additional MS registries be useful?
Well, absolutely. I did mention that we've been very fortunate to collaborate with several groups, academic institutions, and registries to get access to some of these samples, and we're always open to having those discussions and accessing more samples. I think anyone who's doing this work knows that there's a real challenge. There's a scarcity of well-characterized, richly phenotyped samples by which you can do these type of studies. So we welcome anyone who wants to reach out and have that discussion. We're in discussion with several others as well to broaden the number of cohorts that we are investigating in our studies. And I would greatly appreciate whoever asked that question to reach out to me directly. And I'd like to continue that discussion offline. We're very open to collaborating, academic, pharma, centers of excellence across the board. That's our goal here, is to expand the number of samples and provide as much value to as many different types of collaborators as we can.
Thank you, Farhan. So another question is, so someone thanks you and congratulates you. I have a question regarding the selection of MS samples. So I think you touched on some of this, but maybe you can expand further. So
Sure.
for the MS sample selection, there were all the MS patients undergoing similar, or actually I think it's the same target molecule for treatment, or different mechanisms for selecting the appropriate protein biomarkers for selection of the 21-plex panel?
Yeah.
And I'll let you go ahead and answer that. There's a couple more questions, but I'll let you go ahead to answer that.
Sure. In all of our studies, there's variability in the DMT or in the MS treatment that the patients were on. So we haven't done any studies yet that are restricted to only a single treatment or a single mechanism of action for maybe a drug class. That's on our roadmap. We're very interested in characterizing the assay from from that type of utility as well. But with the studies that we had access to, there was various therapies that the patients were on. Now we do track that information. We do account for that information in our modeling as well. We looked for associations relative to subsets of samples that were only on a certain therapy, but none of our studies to date have been specifically designed to address only a single therapy.
Thank you, Farhan. So we're just at the hour. Thank you for the time you've taken to share your research and your findings and the methodologies you've gone through using Olink. Definitely all very interesting. So this is to the attendees. There is a 3-question survey. If you click survey in the control panel, we'd really appreciate you taking the time to answer that. And that just helps us to continue to pump out impactful content. So you'll also be receiving a link with a recording in about a week or so. Again, thank you, everyone. Farhan, thank you for the great presentation.
Thank you.
Everyone, take care.